The following, I am going to present to you for the first time the history of bioindication in the Americas, and especially the bioindication in Latin America. The first thing, why macroinvertebrates? We use macroinvertebrates as indicator of water quality because of their size, as we can see there at a glance. We don't need any special device to see it in the field. They are relatively easy to sorting out. In other words, they are easy to identify. The heterogeneity of the group and his abundance, they are very abundant and the same thing in all parts of the world. And they have long life cycles compared with other species. Long life cycles means 3, 4, 5 months instead of a few hours in some microorganisms. What is bioindication? This technique uses living organisms through which scientists can read the state of pollution of aquatic ecosystems. This is, in other words, bioindication means that if you know where a specific organism lives, you can find it anyway if the water is good or the water is bad, according to the circumstances. But only in water, in clear water, live some specific organisms, and in bad water, worse water, live another organisms. So this is why bioindicators. We can do, or we can tell about the same thing in the different ecosystems. Where do the macroinvertebrates live? They live in the bottom of the rivers. On the rocks and stones, they are buried in the mud and sand and stuck to the shore vegetation, submerged vegetation. We can see over there here some of these organisms, macroinvertebrates. Most of them are larvae from or pupae from insects that the first phase of life is in the water, and then they go through adults later on. So we have most of them are insects. Some of them are also crabs, and we have also some snails. So we have all of this bunch of macroinvertebrates in the water serve for us, or you use for the indication of the water. What is the method of BNWP? The Biological Monitoring Working Party was established in England in 1970 as a simple and a rapid method for assessing water quality using macroinvertebrates as bioindicators. The reasons were mainly economic and for the time required to invest. We need, this is economic, and the time to invest is very short. The method only requires to reach to family level, and the data for the qualitative, and the data is qualitative. This is just to say presence or absence. Scores range from 1 to 10 according to different groups' tolerance of up to contamination. And the sum of the scores of the families give the total score BMPWP. The higher the final score, the lower the degree of the environmental. That was in the 1970 in England and Europe that it was established, this method. So what happened after that? When I was doing my master's degree in Kansas for 40 years ago, I began to establish this method for Colombia. And what happened was that when I sent the first samples to my professors in the United States, they told me, Gabriel, congratulations, because you are doing a very hard work, but everything is completely mistaken. don't have anything. You are going to classify organisms through the keys from the United States and from Europa, but you have to do your own key. You can imagine, I couldn't sleep for many days, for many times, and I was looking for how can I do my own key. So the scores here, assignment for water quality, this is the scores made for Colombia about 30 years ago. It took about 20 years to make different keys for these organisms. You have here families pointed from 10 to 1, and 10 means that you have the organisms that only lives in a better water quality water, and the bad ones means 1. So what is the type of, through these bioindications, we categorize 5 classes. One class is quality good, and the score range about 150, and the color we use for representing the maps are blue. The class 2 is a condition of acceptable water, slightly polluted water, and we represent by green. The third class is a dubious water, moderately polluted water, and we use the yellow color. And the fourth class is a very critic water, very polluted water, and we use the orange color. And the 5 class are very critic, heavily polluted waters, and we use the color red. So this is one of the representations in which we, after work in the field, we use maps in order to represent the different colors according to the quality of water. In the first map, you can see about 38 stations in we sample, and the other one is only just 2 stations in a smaller area. But you can see the colors, and through the colors, you can identify what is the quality of water. Who were the ones who contributed to the application of this method in Latin America? As I told you, when the people, my professor told me, Gabriel, you have to do your own quiz, what the first thing I had to do was to contact people who worked, and he was working on macroinvertebrates, not only in the United States, but also they have a bunch of information, very good information in the tropics, in the Central America and South America. So I found different professors. I'm going to mention some of them very rapidly. Johann Ilyes, he was my advisor for my PhD in Germany, and he was the first. He spent several years in Peru working with aquatic insects. The first key was made in the 50s and the 60s. And the other professor was Joseph Fickau from Museum in Munich, Zoological Museum in Munich. The other one was George Edmonds, very known in the United States, and he is one specialist, or he was, because he is passed away, and he is from the University of Utah. I visited every one of them in the United States. I brought samples over there, and I discussed what will be the real situation of one of these organisms. And most of them also went to Colombia, and we made many field trips. And they stay for 3, 4 weeks in Colombia, and some of them in my house. Robert Wessel, very known in limnology. Werner Lewis, Ephemeroptera from the University of Florida. William Peters from the University of Florida, also in Ephemeroptera. Minter Wessel, also in Odonata from the University of Florida. Most of them, it was a very, very great group in Florida working on neotropical organisms, neotropical macroinvertebrates. One very known, he still lives, and he is in the Smithsonian Institution from the Trichoptera. He was many times in Colombia working with us, and he gave more than 3 or 4 species recording my name because we found many new species in this trip. Another one was Axel Bachmann from University, Hemiptera from the University of Buenos Aires. And we have also Rodolfo Espina from Universidad Nacional de Colombia and Eduardo Domínguez from Universidad de Tucumán. We have also Javier Alba Tercedor from Spain. He helped us very much in trying to how to analyze bioindication with these new organisms for neotropical regions. Narcis Prat from the University of Barcelona, he spent several weeks and several months with us in the Universidad de Antioquia in Medellin, working with us in different fields. And I visited him also in the University of Barcelona. Another person very interesting I knew in Bolivia was Niels de Pauw, who was also in bioindication, and Nico Nisser from the University of Utrecht in Holland, working with Ephemeroptera. This is Charles Hoge. Charles Hoge was also in Colombia several times. I visited him also, and all of this work took about 25 years to make the first key for Colombia. And Dale Habeck, also from the University of Florida, in Pyralidae or waterflies. And here I have a very good friend. I knew him many times, many years ago, Jose Tundisi. We worked together also in one of the lakes over there, and he showed me for the first time one Ephemeroptera in a cane. Yeah. And also Ramon Margalef, I had the privilege to know him, and I was discussing about all of these things. So, what is the state? This was really a story in which, without these people, we couldn't make the first key for us in America. What is the state of knowledge of aquatic macroinvertebrates in the Americas? The first book I wrote was in 1988, a taxonomy key called taxonomy key called the Guide for the Identification of Macroinvertebrates in Antioquia, Colombia. And from this guide began to spread out this information, and now we have most of the countries working based on this guide, but he has been doing his own guide in every country. Then in 2003, I made another book, Bioindication in Colombia, the method of BMP-WP, and this is the application. The first thing we have to know was what we have and then how to use them. Many of the studies in macroinvertebrates in Colombia that have been produced. Here I have two types of books. Most of these books, and they were some of my students doing his thesis degree for a degree in biology or environmental engineering. There are another publication also in Colombia, and there are Diptera. This is a very special Blephariceridae. It's a group from Diptera in Antioquia, Colombia. All of these publications were made in Los Angeles Museum in California. And we have a curiosity, historical curiosities, looking for the literature. In this term, I found a review. It's a collecting trip to Colombia, South America, the University of Michigan, Ann Arbor, 1918, by Williamson. First collection for Odonata, Dragonflies of Colombia, 1916 and 17, in the Magdalena River. It was fantastic, the description, what he makes about the Magdalena River, but that time, now you cannot recognize the Magdalena River through the destruction we have been made. But this is really a very strange curiosity. So this is what started in Colombia, but I am going to give you a very rapid information about what is going on in other countries. For example, in Canada, I found Canada Aquatic Biomonitoring Network. It was for me very interesting. Note the Aquatic Biomonitoring Network is an aquatic biomonitoring program for assessing the health of freshwater ecosystems in Canada. So there are, but the interesting thing is that when I began to study macroinvertebrates, it was in the United States. So the difference in the climate, in the temperature, in the weather is completely different, and that was the difficult thing to make an adaptation for tropical things. So Canada is very well known also in this information. United States of America, ecology and classification of North American freshwater invertebrates. The United States has a long tradition in the study of aquatic macroinvertebrates. It was there when I found the best specs in the 80s that helped me to build the first key for Colombia. In Mexico, there are also very good knowledge about the macroinvertebrates, but I think in Mexico they have to work a little more about this point. Guatemala has a very good knowledge of aquatic macroinvertebrates and developed a lot of recent years. They have been carried out several workshops thanks to Manuel, who is he is very active, and we have done so much great work in Guatemala. El Salvador, they are one of the best one in Central America. Salvador, the knowledge of aquatic invertebrates in Salvador is very good. Already they have their own kits. They have a bunch of books, more than 12 books, each one for one specific group in Salvador. In Costa Rica, I think this is the best one. There in Costa Rica, Monica Springer had developed also a very completely new key for Costa Rica, based on our key, but he adapted and she adapted, and most of them had do the same thing. In Panama, the knowledge is very good. In Panama has helped us very much also, and this is a very no-go knowledge. Honduras have a very good information also, and we found claves de identificación de invertebrados de agua dulce, keys for identification of freshwater macroinvertebrates. República Dominicana, it was for me very interesting to find that in República Dominicana still does not have his own guide by the head study published for preservation of water. There are many regulations I found in República Dominicana. In Cuba, the knowledge of aquatic macroinvertebrates in Cuba has developed a lot in recent years. We had a meeting 1 year and a half ago, thanks to Mercedes, to the help in organizing a special workshop in Cuba, and I was astonished about how much some people know about macroinvertebrates in this country. Jamaica, no records found. Puerto Rico, the knowledge of aquatic macroinvertebrates in Puerto Rico has developed a lot in recent years. There have been also carried out several workshops. There are many people so active in this kind of investigation. Venezuela, the knowledge is also very good, and they have in this moment their own guide. They adapted many books adapted for each country. Ecuador, very good also. I know some people from Ecuador, and they have been working in this theme, and some of them have been in Colombia in my university also working and knowing what we are doing. Peru, very good. I found a very good key. They already have their own guide and many published works. If you look at internet and you look and you write macroinvertebrates from Ecuador, macroinvertebrates from Brazil, you are going to find many information about that. Bolivia, very good. In Bolivia I met Professor De Pau from Belgium, and he has been working over there several years. Especially they have their own key and they use bioindication everywhere. Brazil, the knowledge is very, here I have the Ecología Insectos Aquáticos, but I know there are much more information in Brazil also. This, I am just giving an idea what's going on in every country. Chile, also, there are much information in Chile, and they use in every environmental study used the macroinvertebrates as bioindicators of quality of water. This is why yesterday I told Professor Tundisi that will be very interesting to take in account the bioindication for the quality of water. Uruguay, also, there are the knowledge of aquatic macroinvertebrates in Uruguay has developed already. They have their own guide. I was really surprised to have in Uruguay also information about that. Paraguay, freshwater macroinvertebrates of Paraguay, published by Museo Nacional de Historia. And this is just one of the samples that you can find. Argentina, they have also evaluation of benthic macroinvertebrates as bioindicators or water quality. In North Argentina, have been working also very much. There are some general books also about the Latin America, and one of them is Benthonic Macroinvertebrates of South America, written by Eduardo Dominguez from Argentina. We have, at the beginning of this year, we have an organization, a net, a web in Central America and Caribbean, and we wrote a book about the diversity, conservation, and use of aquatic macroinvertebrates in Mexico, Central America, Colombia, Cuba, and Puerto Rico. This book, it was 4 months ago out of the print. And we are going to have the 3rd Congress in Colombia, in Santa Marta, about macroinvertebrates. This is by the net, or the net from Central America. This is going to be at the beginning of next year. And we have been doing many workshops, conferences, and meetings in aquatic macroinvertebrates. That was one in Bolivia in 1999. Another one, Medellin, Colombia, in 2012. In Nicaragua, 2012 also. In Panama, 2013. In Cuba, 2013 also. And in Guatemala, I want to give the thanks and recognition to Manuel, who helped me very interested in this theme, and he helped to us very much in this theme. So also, in conclusions, the method of assessment of water quality with macroinvertebrates as bioindicators is widely used in Americas, where each country has made its own adaptations. The method is fast, inexpensive, compared with physicochemical analysis, and very reliable. So this is the things I wanted to present to you in today, and I think I am really very proud, because we can think we had made a school on macroinvertebrates in the whole Latin America. Thanks so much.